Well, hello there, ladies and gentlemen. It's Mr. Bleeker, and we're back again. This will be our second lesson in Chapter 25 in the study of DNA. What we're going to look at today is we're going to look at its replication. How do you make clones of that big, fancy, twisted DNA molecule? So let's get started. Head on down to the fourth topic here. We've done the history and structure of DNA, and check out the semi-conservative replication of DNA. When you go there, you'll be here at section 25.2. Of course, this mirrors the existing textbook that we're using, but nonetheless, this is the order of the topics I would always go in anyways, regardless of the book. When DNA of the molecule is replicated, it's very important to clone it as exactly as possible. Why? Because that's the genetic code. We want the same daughter cells to have the same genetic sequences, the same genes. We want to make sure that things throughout our body have consistent genomes. So there's a process that you might have studied in the replication of DNA, and it was called mitosis. It involves chromosomes, cells dividing, and the creation of daughter cells. We're going to zoom in even further on that process, and we're going to look right down at DNA replication. I have a fabulous animation for you today. This animation will also be on the course site, and it's very worthwhile going through and getting to know DNA replication because all parts are testable and you need to know it all. Okay, so we'll get going here. Let me pop up a reflection and we are good. Okay. Give it a moment. Excellent, there we are. So when last we met and we were going over DNA, I'll just open up the shelf here on the side, we talked about its discovery. And we'd gotten to about this point. And I just want to make sure, let's get our friend here. There we go, full screen. You notice that the molecule itself is a twisted ladder. We call that a double helix. But in order to get at it, to read it, and even to replicate it, it has to be unwound. And that this slide 17, for all intents and purposes, shows the unwinding of the molecule so that you can see the hydrogen bonds between the purines and the pyrimidines. The purines, the AGs, and the pyrimidines, the CTs. And we learned about what pairs with what, adenine with thymine, cytosine with guanine. Now we're going to move a little further in this series, and we're going to talk about DNA replication. When your cells are constantly repairing themselves, they're reproducing asexually. So you're supposed to get clone cells. We're not trying to create new cells. We're just trying to replace what's already there. Now the molecule itself also has to be replaced. So it needs somewhat of a template. Because if you're going to make an exact copy of something, like imagine going to a photocopier, you put down the original sheet and then you make a copy of it. Well, that's not totally dissimilar from what we're going to do here, but there's some major molecules involved. And in fact, I see that one's missing. I'm going to write it in. Because DNA is such a twisty molecule, we're going to introduce an extra one here. We're going to look at one called gyrase. Now, gyrase's role is to actually do, we'll say, untwist. There we go. Untwist the DNA helix. Because DNA is fairly, it uses the twist to compress the overall size of the molecule. It becomes very, very small. Remember, we're talking nanometers between each base pair here. And the reason for that is the twisting of the ladder. And there's a demonstration, I have a model in class, and I show when I twist the molecule upwards, it becomes very short. If in computer science we could get files that compressed, it would be absolutely amazing. DNA did it first. So gyrase will do the untwisting. We'll look at another enzyme. These are all enzymes. And how do I know? Well, in true fashion, a lot of enzymes end in ASE. There we go. So many enzymes are going to be involved. We talk about gyrase for the untwisting. Helicase It unwinds the DNA strands by breaking the hydrogen bonds. Think of it as a wedge going through a log, or traditionally it's also been considered to be like a zipper lug, pulling the two halves of the zipper apart. That's a very good analogy. We'll look at enzymes that make polymers, and the polymer we'll look at the several different forms. DNA polymerase, which takes nucleotides and builds them in, and as it does that, we get polymers of nucleotides, and that's what the DNA strand is. When all is said and done, we have to seal up the DNA strand, and that's the role of DNA ligase, sort of like an enzyme applying glue and healing the molecule. Okay, so now we know our players. I'm going to pop out of reflection. One moment. Go. My iPad needs to charge anyways. And seems every day at this exact time my phone rings. Oh well, we'll ignore that. And we'll get to the basics of DNA replication. Okay, so a really great animation, a great course site here. And let's get to, where was I? Here it is, the whole picture. That's what I wanted. So we'll take this in stages. And there's a couple quid pro quos here that you have to be aware of. So I'll go stepwise for this lesson. The first thing that needs to occur is you have to unwind this double helix. Okay. Otherwise, you can't get at the base pairs in the middle. And you can see a little bit of that is already happening here. So for the purposes of replication, we'll send in DNA gyrase. Okay. And if you've ever been on a gyrotron, you sit in the seat and they spin you in all directions. It's an easy term to remember. So let's unwind the helix, and we'll send in DNA gyrase first. Okay. So DNA gyrase is sort of the point man in the whole operation, unwinding the molecule. And in order to replicate it, we have to replicate two parental strands. So this is the 3 to 5 prime strand, okay, called the leading strand, and this is the 5 to 3 here. Now, as the zipper moves down, by the zipper what we're talking about is helicase, it requires energy. So that's why this annoying ATP molecule keeps showing up here. It's supplying energy to the enzyme to break the, albeit weak, hydrogen bonds. Let's push that a little bit forward ahead. There we go. As the lug pulls the DNA molecule apart, and by the lug I'm talking about helicase, the DNA strands need a little bit of stabilizing. So you get these little single-stranded stabilizing proteins. And just go over them with your mouse, you'll see it provides a little bit more information about them, their identity. That's why I like this animation so much. As they provide reinforcement, we're just about ready to start replicating the molecule. Now, the molecule itself, you can't just start replicating the nucleic acids. You can't do that. There's a little bit of a primer required. Now, what's interesting about the primer is you lay down with an enzyme called primase little RNA strands. So we call them RNA primers. And think of them as little focus points for DNA polymerase to begin doing its work. Start replicating here. So you've got little RNA primers, little targets. And it's going to stop and quiz us. So what type of molecule is the primer in blue? What's that thing? Well, it's ribonucleic acid, DNA's cousin. Single-stranded thing. Now we'll focus on the top part of the molecule, and then this is called the leading strand. Now by leading, it grows towards what we call the replication fork in this direction. Think of that as aiming for where helicase is destined. And we're heading from the 5 to the 3' direction. And in fact, you have to know that. DNA is replicated 5' to 3', not 3 to 5. That makes for an interesting problem on the lower strand. We'll get to that in a minute. Now, there's different species of DNA polymerase. This is DNA polymerase the third, And it has a relatively easy job because it sets up aside our RNA primer, and with the addition of nucleic acids, it just grows a complementary strand along the original parent strand. They call this semi-conservative replication of DNA for a reason. The reason it's semi-conservative, semi meaning half, is we keep or conserve half of the original parent molecule to make the new one. So every time DNA divides, or is replicated, I should say, the one half of it is the original parent strand, and what's grown in on the other side is the new strand, or the complement. All right, let's move this forward. Okay, so that red line represents the newly added DNA, and it's complementary to this strand. Now, by being complementary, what you have to realize is these nucleotides right here are exactly the same as the ones down here. They have to be the same because they have to match. So if that was adenine, that would have to be guanine. If that was cytosine, that would have to be, or sorry, let's back up. If that's adenine, that's thiamine. And if that's cytosine, then that would be guanine. So it follows sort of the rules of the DNA alphabet. The replication fork here is, of course, in the 3' direction. Remember, this goes 5 to 3. So nucleotide addition is relatively straightforward on the leading strand. Now, on the lagging strand, it's a little bit different because you notice that the parent here, this is the 5' end. Now, growth always happens towards that. So if you follow my cursor, growth has to happen towards the 5' end, but think about it, we have to grow in this direction, which is, in this case, for the lagging strand, this is a little bit of a problem because this is the 3' end right here. So we have to grow from 5 to 3. So we have to grow this way, then we have to grow this way, and then we have to grow this way. It's done in little installments or in sections. Let's watch that. All right. So press play. And I think I just want to rewind a little bit. Let's go back one. Okay, I'll start from here. And you can see, there we go. You can see that the growth proceeded from 5 to 3, but it was done in little sections. Nonetheless, even if it's sort of grown towards the left, it's starting on the right. Overall growth is still proceeding towards the replication fork. It just, you have to keep laying down additional primers, and then you'll have to grow in this direction constantly in sections. That's really all it is. Now these get a special name, these little fragments here that grow off of the RNA primer are called Okazaki fragments. And you'll notice that you see them on the lagging strand. That's because growth goes in little chunks. So now I'm ready to press play. Answer that question again. I can't get anywhere without it. We'll look at the top strand now. What had happened as the nucleotides were added in by DNA polymerase III is growth occurred towards the replication fork. Now another cousin, DNA polymerase I, comes along, and watch what it does. We'll just press play now, and you can see that DNA polymerase I grows in the DNA over top of these primers. Primers, you might have heard of your parents when they paint their walls, and sometimes they start with a primer, but you paint over the primer. You need to lay down the actual paint. It's no different here, where DNA polymerase I has to grow in the remaining DNA over top of the primers. You can't leave the RNA fragments. Okay, they have to go. So that's DNA polymerase I's job. So the Okazaki fragments, quick little quiz. Where do they form? Well, they're not on the leading strand. You don't need that, right? They're on the lagging strand. Nice. Okay. So as the fragments here on the lagging strand are built together, we have like little gaps here. And we need to fill those in. So DNA ligase, its general purpose is to be a ligand. So if you've ever had stitches applied Those stitches in medical terminology are called ligands. And ligands, glue, stitches, you'll see what it does here. Using a bit of energy, DNA ligase comes in and seals the molecule. And it takes quite a bit of energy here, so we've depleted adenosine triphosphate of two of its high energy bonds. And just for information's sake, once the phosphate is released, if you're looking at that eye there, it's called pyrophosphate at that point. So these components are going to go back to the mitochondrion for a refresh. Now you've got two daughter strands. And the question is, well, is it semi-conservative? Are they the same as the original strand that we began with? So a little question here. The new red strands, what are they relative to their parental strands? Are they identical? Okay. Now by parental strands, we're talking about the green ones. So are they identical to the green ones? Well, that doesn't make sense because that doesn't follow the DNA pairing of the A's and T's, G's and C's. It would seem to be that they would be complementary. Excellent. And by growing in the complementary strand, you've replicated the DNA. So, nice little animation. I'm going to pop back into Reflector because we have a little bit of node information to finish off. There we go. So we'll mirror. We'll go back to the nodes portion here. So there's our major molecules. There's our players. If it's unclear, go back through the animation. Several times students in the past have told me they've had to go through it and look at it again and again, almost until their eyes bleed, until they get it. And then they still come to me for clarification. Absolutely fine. Have to personalize this process. It can be pretty challenging. Okay, here's a picture of the process broken down without showing our enzymatic players here. So the parental strands are these blue fragments, and as they are pulled apart, right? First DNA gyrase unwinds, and then we we have to break the hydrogen bonds, so helicase goes up the middle. So there's no hydrogen bonds between these. And what we do, using our different enzymes of different versions of DNA polymerase, is we're going to grow in, I was going to say draw in, but in this case we're growing them in. We grow in the complementary strand. Okay, so this complementary strand right here, the C, you notice that's exactly the same as the right side of the ladder. Okay, it's new, so we say this is semi-conservative replication. I'll just undo that. And if we look at the left side that's been copied in to the right strand, you notice that it is complementary, and as a result, it's just like this original side. That's why they call it semi-conservative replication of DNA. You can replicate, but you have to use the parent strands, the blue ones here in this case, as templates. Otherwise, you don't have anything to go by. And we'll keep this short and sweet. By the time semi-conservative replication is done, you have a mixture of old and new, and you can see the nucleotides growing in. And they use the base pair sequencing is exactly how this is monitored. And, well, more or less checked for errors. DNA polymerase, should point out, our friend DNA polymerase has a spell checker built into it. DNA polymerase will ensure, that's right, spell check. It will ensure that the A's go with the T's and that the C's go with the G's. It's smart, and if it makes a mistake, that enzyme can back up and fix its mistakes. And it's the reason why DNA is a highly conserved molecule. Now, admittedly, over time, little mutations, little mistakes can happen, but DNA polymerase is like a librarian. It's on the job and it's checking for errors. Not to say that some don't rarely happen. That's why evolution is a very long process, by and large. So having DNA polymerase with that intelligence, seeming intelligence, is a good thing for our genetic code. Viruses that use DNA as their genetic code, for example, polio, you can rely upon those viruses to be the same organism generation after generation. It's why the polio vaccine is something that's been quite stable. We're immunizing against the same kind of polio. Now, that's when a virus has a DNA core. Now, if you remember from Biology 11, when we talk about RNA viruses, you can throw that rule right out the window. Because when RNA viruses replicate their genetic code, they're not replicating DNA, of course, they're replicating RNA. And all sorts of spelling mistakes happen. Okay, there's no the enzyme that does that replication is not on the job. It doesn't check for errors like DNA polymerase does. DNA polymerase does an excellent job at what it does. It's the reason why vaccinations to DNA viruses are much more successful than our RNA viruses. They mutate like heck. So just finish up here. DNA repair, there's the proofreading mention that I made just in the previous slide. And not only polymerase, I'd forgotten to mention that DNA ligase also helps to check for mismatches. It's really the last player in the game that seals up the molecule. So between the two, you can think of it as a librarian and a librarian's assistant. Hey, everything is neat and orderly. Okay, I will discuss DNA organization in our next lecture, but for now, we'll keep it short and sweet. That was the semi-conservative replication of DNA. Thanks for listening, folks.